Yeah. So all very good afternoon, and you don't need any intro, I know that. So yes, before getting into the actual chapters that we are going to deal with from biology, let us once figure out like what are the major highlighted chapters that we have to start with the journey, start our journey with. As NEET 2027, if you are aspiring to, that is because of so much perseverance and resilience you people have after writing NEET 2026 and re-NEET 2026. So now let us look into the chapters that we are going through for NEET 2027. Considering your past 10 NEET papers into consideration, taking your past NEET 10 papers into consideration, let us look like how often you got questions from the chapters. If we have to see first from living world, let us have a blueprint and then we will go on the concepts. Living world, if we have to talk about NEET 2024, 25 or previous papers, if you see, hardly used to get one question or no question at all from living world. But then now if you observe in re-NEET 2026, you got two questions from living world, right? One is statement based and the other is match the following from taxonomic categories, right? So that is about living world. In living world chapter, what are the concepts you have to revise more or practice more this time based on my knowledge? Defining and characteristic features of living, you can expect some statement based questions and all. Most important highlighted concept is taxonomic categories. All the 7 obligate taxonomic categories and subcategories we have. That is the major hotspot of this chapter. Next is taxonomy and binomial nomenclature. Binomial nomenclature and taxonomy. Defining and characteristic features of living also. These are the major areas that you have to focus from this chapter. And then coming to biological classification, starting from systems of classification, from 2 kingdom to 6 kingdom system of classification, then kingdom Monera, Protista, fungi. From fungi definitely there will be a question, mostly very often you can see when kingdom Protista, Monera, fungi are considered, more often you can see questions from fungi, especially from the classes of fungi, reproduction of fungi. Okay. So that areas you have to focus. Every year maximum you may get 2 questions from biological classification, either 1 or 2 questions from biological classification, and it has of 7% weightage from the total chapter, and this is of living world, minute weightage. And then is the chapter plant kingdom. Okay, plant kingdom. Plant kingdom also hardly you will be getting around 2 to 3 questions. If we take all the 10 papers, 10 old papers like from 2017 to 2026, if we have to say, every year you can see 2 questions or maximum 3 questions from plant kingdom. Like you may get questions from gymnosperms very often. In NEET 2025, if you see, same pattern question asked from bryophytes and pteridophytes, multi-statement question on life cycles. Both bryophytes and pteridophytes, same pattern of question, just they changed the name of bryophyte and pteridophyte, multi-statement based questions. Like that you may get it, and most importantly from algae, chlorophyceae, phaeophyceae, rhodophyceae, from all the 3 different classes of algae also you got the questions multiple times. So those are the areas where you have to focus for plant kingdom. How many questions you may get? 2 to 3. It costs across 9% of the total weightage. If you have that NCERT 11, open the index paper and listen to the class, look at the concepts and weightages. Okay. Next, animal kingdom, zoology sir will be dealing with it. And coming to morphology of flowering plants, usually this chapter will be starting with vegetative characters, but we people, after learning morphology for all the 2 years, we have to be clever enough to start with families, because if we are starting with families, there only vegetative characters will be covered. floral characteristics will be covered. After families, we have to come to what? Floral characters, where we have to learn on flower types based on the position and symmetry, aestivation, placentation, types of ovules, all this. Okay. So, that is how we have to go with what? Morphology. And since the syllabus have changed from NEET 2024, they have included families. But then we got a one question in NEET 2026 from families, that too from Fabaceae. Okay. Likewise, we will start first from families and then floral characters, and then we will move to vegetative characters. What is the level of questionnaire you will get from morphology? 2 to 3 again. Okay. Maximum 2, pretty maximum 3, maximum to 2 only. Then comes anatomy of flowering plants. The beauty of this chapter is anatomy of flowering plants is important for IP, it is important for MCET, and it is important for NEET as well. Here also from this chapter you will get 2 to 3 questions, okay, wherein you have to focus majorly on tissue systems. Okay. If you are beginners, then I will be starting from tissues, then tissue systems, then comparative analysis, and then secondary growth. But you people are experienced, right? So, pretty well we need to start with what? Comparative analysis between dicot, monocot, root, stem, leaf. Okay. We will start with the concepts which are highly important. Okay. So, every year if you observe 2 to 3 questions, maximum 2 questions from anatomy we will get. One question will be definitely from comparative analysis of dicot, monocot, root, stem, leaf. From either of the 3 you will get one question. Very often we saw questions on stem, that too on dicot stem. That is about anatomy of flowering plants. Next, cell the unit of life, the chapter which we are going to start with. This chapter 2 to 3, 3 to 4 questions also you can see. If you observe NEET 2026, we have seen the paper discussion. Hardly 4 to 5 questions I can see, like mostly 4 to 5 questions are from cell the unit of life only. And this chapter, every time we will start, we will begin with this chapter, because this serves as a starter. Okay, to enjoy this complete menu, the starter will be cell the unit of life. If you know completely about a cell, prokaryotic, eukaryotic cell, then you will understand the dynamics of the other chapters as well, be it core botany, zoology or physiology. Okay. And then comes biomolecules. Very important to remember, like we saw many questions in the biology where the question is not just asked from a single chapter. From 2 to 3 chapter a single question is framed. Likewise, if a question is framed from cell the unit of life in combo, the combo can be sexual reproduction of flowering plants, it can be genetics most probably. Okay. From mitochondria and chloroplast, if they are picking up mitochondria and chloroplast, then the combo can be plant physiology, respiration in plants and photosynthesis in hive. These are the combo chapters where you will get the common questions. Got it? And then biomolecules. Biomolecules also 2 to 3, sometimes 4 questions also, very important and vast chapter. From biomolecules, there is a concept where NTA will never escape even a single year. Can you tell me that concept? From biomolecules, like major concept of biomolecules, if we have to imagine biomolecules as a movie, the second half completely will be that concept only. Enzymes. Enzymes, very important concept where you will be getting it every year from enzymes. And then comes cell cycle and cell division. Cell cycle and cell division, you will hardly get 2 questions, and mostly application based questions are from cell cycle and cell division. If you see NEET 2026 also, you got a question in connection with sexual reproduction of flowering plants, because mostly cell cycle and cell division questions will be linked or connected with ploidy, and they will be asking you the questions like diploid, haploid, connecting it with mitosis and meiosis, they will be asking you questions. Mostly the combo questions will be with sexual reproduction of flowering plants. Got it, right? So, this is about cell cycle and cell division, right? Application based numerical approach type of questions you will see here. Then comes plant physiology. From that, first and foremost major chapter is photosynthesis in higher plants. Okay. So, all these chapters from biological classification, plant kingdom, morphology, anatomy, which are core botany chapters, we may expect 7 to 8 questions on total core botany chapters. Now, coming to applied concepts like cell biology and plant physiology, from this put together you may expect around 10 to 11 questions. Okay. You can draw out also from the, if you are not trusting my words, you can draw out also considering all the PYQs into consideration. Here if we see photosynthesis in higher plants, from photosynthesis rarely we got questions from discoveries, but previously discoveries part also asked in a different way in NEET 2026, right? Yeah, we never expected that, but then NTI surprised us, but we need to learn the subject in that way also, like how the discoveries have started, starting from bell jar experiment of Joseph Priestley, first about air and then about oxygen, then Jan Ingenhousz said about oxygen is evolved from photosynthesis, then lastly it is E.W. Engelmann who gave us action spectra and absorption spectra. Like that, this time we need to learn on the order of discovery also. So, and then mostly questions every year you will get from light independent reactions, that is C3, C4, CAM cycle, and 2 questions are from photorespiration in NEET 2026. Did you see that? 2 questions are from photorespiration, C2 cycle. Okay. So, from these 4 cycles you will get the questions, mostly from bioenergetics of C3 and C4 cycle you will get the questions. Next is respiration. Glycolysis important. After glycolysis you may get the every concept from respiration in higher plants is important only, right? Glycolysis, anaerobic respiration, Krebs cycle is important, ETS cycle, amphibolic pathway, respiration coefficient. There is not even a single point where we can ignore from this chapter. Next is plant growth and development. Like how from biomolecules, second half we said enzymes is pretty well important, every year you will see a question. Like that there is a concept from plant growth and development also. Very good Harshita, very good. So, definitely every year there will be a question from PGR, plant growth regulators, okay, like synergistic antagonistic functions of different phytohormones, which phytohormones are promoters, which are inhibitors, about their discovery, about their precursors, derivatives, every inch we have to look into. Okay. So, this is the complete picture of the NCERT 11, the chapters which I am going to deal with you people for this whole year. Okay. So, you got an idea, right ma? From every chapter, the level of questions, concepts. Look at the pie chart also. If anywhere you are feeling like any add-ons should be given or still you have any question marks on any chapter, you can ask me. If you are okay, we will move to NCERT 12 concepts. Okay. Yeah. And now, from NCERT 12, the chapters that we are going to deal with, that I am going to deal with are just 4. But those are broad and major chapters, of which 3 are broad and major chapters. First, sexual reproduction of flowering plants. This chapter itself will cover around 3 questions. This chapter itself singly it will cover around 3 questions, 2 to 3 questions. But in combo, the combination can be with plant kingdom, the combination can be with morphology of higher plants, the combination can be with cell cycle and cell division. With these chapters, any combo questions you will get, that can be around 2 to 3 again. Alone it will, you will have 3 questions likewise. With combo chapters you will have again 2 to 3 questions. Then comes principles of inheritance and variation and molecular basis of inheritance. These 2 chapters, broad chapters, vast chapters where you will enjoy biology here. Okay. Starting from heredity in class 11, 11th, 12th and here also we will be learning, and this continues even in your graduation. Learning it properly is very important. Okay. not just considering your NEET exam, but higher standards as well. So, here if you observe principles of inheritance and variation, from here hardly you will get 2 to 3 questions again. You mostly, if we have to see the previous year questions, either from dihybrid cross, linkage and recombination, sex determination, we can see the questions mostly on the chromosomal disorders and genetic disorders. These are the areas we need to focus on from principles of inheritance and variation. Next is molecular basis of inheritance. If we see the number of questions from Mol Bio have increased from the year 2025. Before that it was around 3 to 4 questions, but from NEET 2025 we can see 5 to 6 questions just from this chapter, molecular basis of inheritance. And again chota sa chapter, microbes in human welfare, like how we have living world in the NCERT 11, here we have microbes in human welfare, where you have to remember about microbes, pretty small examples, memory based, where you may get hardly 1 to 2 questions from microbes in human welfare. Got it? This is the chapter description according to your chapter wise weightage considering past 10-year NEET papers. Okay. Now we are going to start on with cell the unit of life. Yeah, just a second, ma. Is the voice audible now? Ankita? Yes. Just give me few moments. Yeah. So, from cell the unit of life, any chapter we need to learn it concept by concept, exercising all the previous year questions, all sort of possible questions, be it statement, match the following, assertion reasoning, multi-statement based, all sort of questions we will be solving after every minute concept. Okay. So, here if we have to fragment this chapter, we have discussed it already, but for new students I am telling again, cell we have fragmented it into 3 parts. Initial session we will be learning on cell discoveries. Okay. Good afternoon. So, initial first part we will be learning on cell discoveries, cell theory, prokaryotic cell. Prokaryotic cell or cell theory, every alternate years you will see one question. Definitely, every alternate years there is a one question on cell theory and prokaryotic cell. Apart from your NEET, to learn on prokaryotic cell is very important. If you have to know the dynamics of microbes, okay, in your graduation might be in further standards, learning on prokaryotic cell is very, very important. Next is differentiation between prokaryotic and eukaryotic cell, after which eukaryotic cell you will start on learning with. First, we have 2 types of eukaryotic cells to learn here. What are they? Plant cell and animal cell. Plant cell, the outermost limiting boundary is what? Cell wall. So, first we will finish off learning on cell wall, after which we will be learning on what? Cell membrane. Every alternate year again you have a question either on the cell wall, mostly on the fluid mosaic model. Every alternate years, like 2 to 3 years, if you see, there will be one question on what? Cell membrane. Okay. And then comes about organelles. From organelles, if you see, beta, now we have fragmented the learning of organelles, we have fragmented into 3 parts. First, non-membrane bound organelles, single membrane bound organelles, double membrane bound organelles. Non-membrane bound organelles, first we are learning on what? Ribosomes. So, questions here from ribosomes, you will not just get from cell the unit of life, you may get the questions from asking about polysome, you may get the questions on ribosomes in connection with translation from molecular basis of inheritance. So, from ribosomes, this is you have to learn this in a broad spectrum, chapter by chapter, everywhere you will hear this term ribosomes. It has only one function, that is protein synthesis, but then in multiple ways it will perform. Okay. So, in broad spectrum you have to learn on ribosomes, because every question, every NEET question paper you will see this term either in options or in questions. Next is centrioles. Centrioles also, if we have to consider our past 10 papers, you got 2 to 3 questions on centrioles. Okay. So, this is about non-membrane bound organelles that we are learning. Next, about single membrane bound organelles. Single membrane bound organelles, learning about this, we will also learn on what? Endomembrane system. Okay. Endomembrane system includes those single membrane bound organelles whose functions are coordinated. Very good. You remember it? Nice. So, here we have endoplasmic reticulum of 2 types, smooth endoplasmic reticulum and rough endoplasmic reticulum, and then Golgi bodies, lysosomes, vacuoles. If we are considering an animal cell in which the vacuoles are of primitive structures, there sometimes we need to consider ER, Golgi bodies, lysosomes itself as endomembrane system in animal cells. Along with this, we also have what? Microbodies, single membrane bound organelles. We have 3 types of microbodies. What are them? Glyoxysomes, spirosomes and peroxisomes. Peroxisomes you will be learning again in plant physiology when you are learning on photorespiration. Then comes about double membrane bound organelles. Learning this double membrane bound organelles crystal clear here itself is very important. You should know all the structural components, all the functionality with respect to each organelle here, because learning mitochondria here clearly will help you in proper understanding of aerobic respiration when you are dealing with respiration in plants. Learning on plastids here clearly. Plastids, of course, in plant cell we have 3 types of plastids. Do you remember? 3 types of plastids. Leucoplast, chloroplast, chromoplast. Okay. Leucoplast, chloroplast and chromoplast. These are what? 3 kinds of plastids, of which chloroplast you will learn here in detail. Learning on chloroplast here detail completely on the structural components, it will help you in proper understanding of sites of reactions, both in light reactions and light independent reactions. Sites of reactions are very important. Next, about nucleus. All the 4 components of nucleus you will be learning, and inside nucleus what is present? Genetic material. Genetic material is mostly in the form of chromatin, but then when cell division is activated, you can see chromosomes at the end of prophase. Clear structure of chromosome is visible. That chromosome structure and shapes you will learn at the last of this chapter. And in this, learning on the microtubular arrangement of cilia and flagella is very important. If you see all the 10 PYQs, 10 previous year questions from this chapter, 2 to 3 questions you can see diagram based questions from cilia and flagella. The microtubular arrangement of cilia and flagella, diagram based questions you can observe. Next is on cytoskeleton, where you will learn clearly about microtubules. If you want this flowchart, you can take in your notes now. Microtubules, microfilaments, and intermediary filaments. Right? If this flow chart is with you when you are dealing with the chapter for the first time, for nth time before your revision, you can track on your concepts easily. Okay? So, this I can say for the students who are listening in the virtual platform as well. If you want, you can take a screenshot of this flow chart now and later you can copy. Okay? Right? Augustine, you are noting down, right? Yeah. Take 2 minutes of time, both students who are listening in the virtual platform and here. Look at it. If you want to take a screenshot, you can. If you want to note it down, you can. So here, starting with this term, very basic term that we are learning from not childhood, around 6th, 7th like that, that cell is the basic structural and functional unit of life. But then if we have to look into the hierarchy of living organization, how a single organism is built up, if we have to look into the hierarchy of living organization, it starts from atom. Group of atoms will form up what? Molecule. Group of different molecules will form up what? Macromolecules. This macromolecules, if they have carbon embedded in it, they form what? Biomacromolecules. Can you name few biomacromolecules? We have learnt in biomolecules chapter, right? If you remember, few biomolecules: carbohydrates, proteins, lipids, amino acids are the building blocks of proteins. Proteins, carbohydrates, nucleic acids, lipids. These are few biomolecules that you have to remember. So those pile up and again they form into what? Organelles. Like just now we have seen non-membrane bound, membrane bound, single membrane and double membrane bound. Then they form up what? A single cell. Till here there is a teamwork. If they are in a team only, they are recognized as a structure or as a function. But here cell is independent in its structure. Single-handedly it can handle everything. It is making a separate structure and it is performing all the essential functions. You can consider metabolic functions which are required to keep the cell active. Single cell itself is performing that. What is the live example? Unicellular organisms. All the unicellular organisms are surviving. They are alive because they are independent in their structure and function. That is the reason we got this popular statement that cell is the basic structural and functional unit of life. Learning about the minute detail of each statement from NCERT will make you more efficient in answering assertion reason questions and statement based questions. Okay? So always this time when you are revising or relearning biology, keep the questions why, what, how, where. These 4 questions are important. Why, what, how, where. Okay? How is it happening? Where is it happening? Why is it happening? You have to know. Next, group of cells. What kind of cells will form tissue? Again, this definition is important for us when we are learning histology and anatomy of plants. The definition of tissue is very important. Any group of cells which have common origin and common function, but they can vary in its structure. Any group of cells with structural similarity or variability, but then they have what? Common origin and common function. Such group of cells will form up what? Tissue. If you get 2 statements, group of cells with common origin and function, mandatory structural similarity, what you will choose? Statement 2 is false. Structural similarity can be present or not. That is how what type of tissues are formed? Complex tissues. In plants we have seen, right? What are the 2 types of complex tissues? To the students in virtual platform also, what are the 2 types of complex tissues? Simple tissues we are learning parenchyma, collenchyma, sclerenchyma. Now you say complex tissues. Yeah. Skanda just has to open up, that's all. She has all answers but she will keep it with herself. That's it, Ray, you say for me. Yeah, xylem and phloem. Xylem has 4 different types of tissues, right? What are they? Tracheids, vessels, xylem fibers, xylem parenchyma, phloem sieve tubes, companion cells, phloem fibers, phloem parenchyma, this and all. So, such will form a tissues. Group of different tissues will form up what? Organ. Group of different organs will make up forming an organ system. Like for example, stomach and different parts of the alimentary canal will build up what? Alimentary canal. This is an organ system. Different organ systems in the body will form what? Organism. This is about what? Hierarchy of living organization. Let us build on some more basics where we can learn on hierarchy of ecological organization as well. Group of different organs will form up what? Group of similar organisms will form a population. Different group of populations will form community. Different group of communities will form what? Ecosystem. Okay, like examples, water ecosystem, marine ecosystem, grassland ecosystem, and then group of different ecosystems will form up what? And then this is the hierarchy of ecological organization. Got it? So this is about a cell, about definition of tissues, hierarchy of living organization and hierarchy of ecological organization. And then if we see about a cell, okay, cell, the limiting boundary of a cell. Now the next, after cell discoveries, we are going to learn cell theory, right? Cell theory, is it applicable to all the type of cells? All the type of cells? No. Cell theory is applicable to those cells which has a cell membrane, cytoplasm, genetic material. Means single nucleus. That genetic material is having cell membrane, nuclear membrane or not is a different story. But then it should have a genetic material, cytoplasm and cell membrane. Here you need to learn on two synonyms also. Might be you already know. There will be some bits and gaps in your mind when we are learning it completely as a chapter again. That bits and gaps should be filled. That is my concern. So here cell membrane and cytoplasm, this together is addressed as cell membrane and cytoplasm together will address it as cell membrane, cytoplasm, genetic material with all the cell constituents. This you can also address as protoplast. Protoplast with cell membrane, cytoplasm, genetic material. Only cytoplasm and genetic material, protoplasm. Protoplasm and protoplast. Clear, Mama, till here? Now, yeah, on cell discoveries. Initially in the beginning of this chapter, about 3 cell discoveries we will be learning. Hardly we will get a question from this area, but we have to know about these 3 discoveries. First, dead cell is discovered by whom? Robert Hooke. From the plant Quercus tuber. Which part of the plant did he discover? Bark of the plant. Where when he have kept the bark under a microscope, he observed the hollow compartments, basic hollow compartments in the form of honeycomb like structure. That is his discovery, and he mentioned about this in his book Micrographia. Okay, hardly if I remember, you got a question in EAP set, but not in NEET, but you remember this discoveries also. Robert Hooke, who discovered dead cell, mentioned about in his book Micrographia, and then he observed this like a hollow compartments, right? And then Anton van Leeuwenhoek, he discovered living cell. Okay, this living cell, how did he discover? Those organisms which are not visible to the naked eye, but then he could see with the lenses. He has made different lenses with different magnifications. He self prepared it. So those organisms which are observed through those lenses but not visible through the naked eye, such organisms he named it as what, Nana? Animalcules. He named it as animalcules, and he is known as father of microscopy because he discovered so many microscopes. And then nucleus is discovered by whom? Robert Brown. He discovered nucleus. He has discovered this in which part of the plant? Orchid root cell. He has discovered the nucleus in orchid root cell. Till here it is clear, right? 3 basic discoveries. Just go through it and then we will start on cell theory. Register in your mind these basic things. Will not keep much focus, but then when question will come from this area, there should not be a regret. We should answer. Ankita, Gaurav, listening. Next, about cell theory. Cell theory in the year 1838. Cell theory in the year 1838. First, it is discovered or proposed by German botanist. He is Matthias Schlitten. Okay. In the year 1838, what did he see? He has analyzed or studied all the plant bodies, and he said all the plant bodies are made up of different kinds of cells, tissues. Okay, they are composed of different kinds of cells and tissues and products of cells he have mentioned. Parallelly along with him, Theodor Schwann in the year 1839. He is who? British zoologist. Many times the statement based questions asked British zoologist, German botanist on these terms. If after learning the entire story of all chapters, few questions will be confused with these terms again. But no, these are pretty basic concepts. We will register it here itself. First, who proposed in the year 1838? German botanist, and then British zoologist in the year 1839. He is Theodor Schwann. Though he is a British zoologist, he is the scientist who said that the limiting boundary of an animal cell is cell membrane. Over that there is one more layer is there in a plant cell, that is cell wall. This statement asked around 2 to 3 times in your previous year NEET exam, around 2 to 3 times it is asked. Okay. Though he is a British zoologist, he mentioned about cell wall as a limiting boundary of what cell? Plant cell. Clear about this? Any doubts anywhere? Yeah, these are the points you have to remember. Of course, they have studied plant body, they have studied animal body, but they failed in saying one point. What is that? How the new cells are formed. How new cells are formed, they failed to say. That is compensated after a long back, see from 1839 to 1855. It is documented and said by Rudolf Virchow that all new cells are formed from pre-existing cells. New cells arise from pre-existing cells under the concept omnis cellula e cellulae. Okay, under the concept omnis cellula e cellulae. What does this mean? New cells arise from pre-existing cells. See, here from this slide what you have to remember is the years of the discovery, scientist names, what are the points they have proposed, and what is the discovery of Rudolf Virchow. Questions about the omnis cellula e cellula will be asked under the heading of modified cell theory. Under the heading of modified cell theory. Okay. Now, again a question. Cell theory is applicable to what type of cells? Just now we have discussed, right? Any cell with a cell membrane, cytoplasm, genetic material. That genetic material may or may not surrounded by a nuclear membrane. Cell theory is applicable to prokaryotic cells also. Okay. So, that is the primary condition on the application of cell theory. When that is considered, we have to look into all sort of exceptions of cell theory. Okay. What are the exceptions of cell theory is very important. Please take down, ma. Exceptions will not be there in your text. But questions will be asked, because about all the organisms that we are mentioning here, na, they are there in your NCERT 11. They are family members of your NCERT 11 chapter, NCERT 12 also you will see. Exceptions of cell theory. First, viruses. Can anybody tell me what is a virus composed of? What are the components of virus? Genetic material, protein code. Very good, Skanda. Genetic material and protein code, but then in few viruses we can see envelope also over the protein code. Can you tell any examples of enveloped viruses if you remember? Examples of enveloped viruses. May I? Herpes virus, HIV virus, All these are enveloped viruses only. So here please note down the components of viruses. It just includes genetic material with a protein coat and an envelope. Okay. Can you see any cell membrane, cytoplasm, nucleus? So cell theory is not applicable to viruses. They are known as living entities. They are not known as living organisms. They are known as what? Living entities, because without host it cannot survive. Next, I have heard about useful bacteria and harmful bacteria, but I never heard about useful virus and harmful virus. All viruses are what? Harmful only. Can I say this? All viruses are obligate parasites.
Yes.
You got a question in the NEET 2026. All viruses are obligate parasites. So, can I name them as purely infectious agents? Infectious agents. You can also address them as obligate parasites. Right? So, first exception is what? Virus. Cell theory is not applicable to viruses. Second point, cell theory is not applicable to enucleated cells. Enucleated cells ante enti nana? Nucleus undu. Those cells in which nucleus is not there, to such cells cell theory is not applicable. If you remember any such cells, you tell me. You have already gone through the complete syllabus, right? If you remember, you tell me. Here. The discussion only you make with me. I will not think anything. What, right? Viroids, prions, yeah. To those also this is not applicable. Very good, very good. But they are again entities only. They are again particles only. Okay. What are those? You said viroids and prions. Good. You can add on this one. Enucleated cells. Can I say matured RBC? Very good. Sieve tube cells, good. And tracheids. Any of the enucleated cells you can see or you will come across in NCERT 11, 12, keep it up there. Okay. Next, third is binucleated or multinucleated or sinucytic cells. Okay, multinucleated or sinucytic cells. You remember anywhere? Those cells which have many nuclei. To such cells also cell theory is not applicable. Can I say rhizopus? Bread mold. Rhizopus or bread mold. Can you remember rhizopus or bread mold is a fungus which belongs to which class of fungi? Very good. Rhizopus, commonly known as bread mold, belongs to Phycomycetes. These are the exceptions of cell theory. Please take down one. Students in the virtual platform, you can take the screenshots. done. Cell theory, its exceptions are done. Now we will come across, before learning different shapes of the cells, we will move on learning with a prokaryotic cell. Fine. We will start learning on a prokaryotic cell. Today we will discuss completely about cell envelope, and in the next class we will enter into the cytoplasm, which is the main arena of all cellular activities. First, considering the prokaryotic cell, we have learnt the limiting boundary of a plant cell is cell wall, limiting boundary of an animal cell is cell membrane. But then prokaryotic cell, which is infecting the plant cell and animal cell, is well equipped with 3-layered cell envelope. Okay, it is well equipped with how many layers? 3-layered cell envelope. Cell envelope which is 3-layered. This 3-layered cell envelope has outermost layer is what, ma? If you remember, yesterday we discussed, right? Very good, Harshita. Glycocalyx. Augustine, you also know. Second layer is? Second layer. Cell wall. Third layer? Very good. Okay. So, we have glycocalyx, cell wall, cell membrane. Glycocalyx, which is the outermost wall layer, it is composed of polysaccharides and murein. Okay, it is composed of what? Polysaccharides and murein. This glycocalyx can be of two types. If polysaccharides are tightly arranged, if it is of this association tightly arranged, this turns up forming what? Capsule. If they are loosely arranged, that forms up what? Slime layer. If they are loosely arranged, they form what? Like this. If they are loosely arranged, it forms up slime layer. What set of bacteria or which group of bacteria glycocalyx will be like a capsule? Pathogenic bacteria, because capsule whose outer layer means thickly composed, densely composed. Like we have when we are learning on transformation experiment of molecular basis on Streptococcus, smooth walled S strain bacteria will kill the rat, will kill the mice, sorry, it will kill the mice, S strain bacteria. That S strain means what? Thick layered capsule. So, capsule is present mostly on what? Pathogenic bacteria. To escape from host immune system. This escapes from host immune system. Okay, this is to escape from host immune system. Next is what? Slime layer, where polysaccharides and mureins are loosely arranged. This is for that set of bacteria which lives in extreme environmental conditions. It can be halophils, it can be thermoacidophils. Okay, to that set of bacteria, slime layer, its outer layer is composed of slime layer. This is to prevent the loss of water and mineral nutrients. Right. So, this is about glycocalyx, which is either capsule or slime layer. Okay. And then cell wall. Cell wall is composed of peptidoglycan. Okay, cell wall is composed of what? Peptidoglycan, which is proteins and glucose units. Proteins will be present in the form of what? Amino acids. Glucose is present in the form of NAG units, which is nothing but N-acetyl glucosamine. Now, based on the percentage of peptidoglycan in the cell wall, peptidoglycan can be 20%, peptidoglycan can be 80%. Based on the percentage of peptidoglycan, we have two types of bacteria. Again, what are they? Gram positive and gram negative. We will learn on it when we are learning on gram staining. Okay, but at here you remember, proteins and glucose units are composed in peptidoglycan, right? Which is in cell wall. It can be 20% or it can be 18%. The percentage of peptidoglycan will decide whether it is gram positive or gram negative bacteria. And then next is what? Cell membrane. Cell membrane or plasma membrane in prokaryotic cell. This will have many invaginations. To the cell membrane itself, the invaginations of cell membrane towards the cytoplasm. Okay, invaginations of cell membrane towards the cytoplasm. Those invaginations can be in the form of tubules, can be in the form of vesicles or cisternae, lamellar, like this. Any of the invaginations towards the cytoplasm, these are known as what? Mesosomes. Along with mesosomes, ribosomes which are present, right, in prokaryotic cell, they are at two locations. Ribosomes present inside the cytoplasm, ribosomes are also present attached to the cell membrane. Larger subunit of ribosome is attached to cell membrane of prokaryotic cell. These are two components which are attached to plasma membrane. Okay, learning on what are the major functions of mesosomes is very important. That we will do it tomorrow. Okay, so tomorrow we will finish off about prokaryotic cell, starting from mesosomes on complete details of prokaryotic cell, and then we will be looking into eukaryotic cell diagrammatic descriptions. Okay, after completing tomorrow's session, we will also exercise the p-vacues, exercise some statement based and match the following in terms of quiz. That quiz is not among you people, it will be with the virtual students also. Okay, let us see who will win, whether those students or you people. But we will have a very nice quiz, active quiz, okay, after tomorrow's session. Fine? Yeah, that's all. Thank you for today's session.